So let's see about this human reproductive system. So before we talk about what is reproduction, it is a fundamental biological process. You can take reproduction as fundamental biological process which will be helpful for an organism to produce the offsprings. So organism producing the offsprings. Okay, of their same species. So why do they are producing offsprings? For the continuation of their species. For continuation of their species. So we call it as the reproduction. Reproduction. That means you are producing the offsprings or the same species of organisms again and again. Okay, so reproduction can be of two types. One is the asexual reproduction, the other one is sexual reproduction. So one is asexual reproduction. The other one, it is a sexual reproduction. So what is asexual reproduction?
Only one parent is involved.
Yeah, only one parent is involved, and whatever the offspring that is produced, it is genetically similar, genetically identical to each other and to the parent. So remember, only one parent is involved. Next one, offsprings, whatever they are produced, these offsprings are genetically similar. Identical to each other and to their parents. Is this clear?
Yes.
Whereas the sexual reproduction, it will involve in the gametes involved in the two parents. Okay, so whatever the two parents they are producing the two gametes, those two gametes will fuse and result in formation of zygote. From that zygote, a new organism is produced. So that we call it as sexual reproduction. So here, two parents are involved. Second thing, whatever the offsprings are produced, they are genetically unique. Clear?
Yes.
One second. So what is the advantages of sexual reproduction? One is, as here it involves a fusion of two gametes. Okay, so one parent, it is bringing its own genes, the other parent, it's bringing its own gene. Those two genes are mixing up here. So there is a mixing of genetic material. So mixing of genetic material. So that's one thing. Second one, as the genetic materials are mixing up, so one parent will be having a set of genes and the other parent is having set of genes. So both the genes together will show different types of phenotypes. So there is a greater variation. And one organism is adapted to different conditions, the other organism, the mother, is adapted to different conditions. Now this father and mother making up the child who can adaptable to both the parents. Yes or no? For suppose father is adapted to certain situations, mother is adapted to certain situations. Those adaptations will also be inherited to the offspring, where this offspring can adapt to this region as well as this region. Whatever the mother got the capabilities as well as the father got the capabilities. So there will be greater adaptability. Okay, so you can see here, there is a variation in the skin color in the different people throughout the world. So what are the secondary sexual characters in male? So first of all, what is the secondary sexual characters? So these are the secondary sexual characters. They will be developed in the individual after attaining the puberty. Remember, so secondary sexual characters developed after puberty. And these secondary sexual characters, they are different in the males as well as in the females. You can see the males are having certain characters where they are absent in the females. Females will be having the unique characters. First, suppose if you see, remember, distinct in males and females. It will be different in males and females. First, suppose as we are talking about the secondary sexual characters in male, first one, you can observe that the larynx gets bigger in the males. So larynx gets bigger in males. So because of this, the voice of the males deepens. So voice deepens. So that's the reason why the males will be having deep voice or the bass voice. Next one, armpit air grows. Penis, prostate, seminal vesicles, these will become bigger. So they grow bigger. And in the males, you can also observe the acne once they reach the puberty. That's the reason why during the 16, 17, 18, there's a more acne formation. And there is a facial hair grows. Pubic hair grows. And in males, the muscles will develop due to the testosterone. So muscle develops. So all these secondary sexual characters are developed due to the male sex hormone. That's a testosterone. The same kind of secondary sexual characters you will be seeing in the females, but they are somewhat different. For suppose in females, you will be seeing the high pitch voice. Actually, the voice is bright compared to the males. In males, the voice is deep. In females, the voice is bright. So breast development, enlargement and development for the milk production. Armpit hair grows. Uterus grows bigger so that the female can accommodate the baby inside. Pubic hair grows. And body shape becomes round. So these are the secondary sexual characters that one can observe in the females after attaining the puberty. Clear?
Yes, sir.
So sexual dimorphism. What is a sexual dimorphism? Di means two. Yes or no? And what is meaning of morphism? Forms. So it is forms. So if you see here, in humans, it shows the sexual dimorphism. That means if you see the morphology and the anatomy, the male and females were different. So there is a difference in the external character features. So you can see the morphological dimorphism. So one is morphological dimorphism and the other one is anatomical dimorphism. In morphological dimorphism, you can see there is a difference in the external features. Whereas here, difference in the internal features. So based on this, the males and females, they are distinct individuals and they will exhibit different characters. So you can differentiate a male and a female. Okay, so let's see about the male reproductive system. If we see about the male reproductive system, it will come two things first. One is the primary sex organ, that's the gonads, and the other one is the secondary sex organs. So remember, the male reproductive system is classified into primary sex organs and the secondary sex organs. Remember this. So what is meaning of primary sex organs? It is very, very important. Yes or no? Compared to the secondary sex organs, primary are very important. Why? These are directly involved in the gamete production. So remember, primary sex organs, which are directly involved in gamete production. That means formation of the male gamete, these are directly involved. Whereas in the secondary sex organs, they will not produce the gametes directly, but they will be helpful for conducting these gametes from the male sex organs to the female sex organ. Okay, so they will help in transportation, maturation, and storage of gametes. So what comes under the primary sex organs? It's the directly involved in the process of gamete production. So they are nothing but the testes. Yes or no? Where does the gamete production will happen? Inside the testes. So primary sex organs, it's nothing but the gonads, or you can say, what are the male gonads? It's nothing but testes. They are male gonads. So testes, one thing they will do is it will produce the sperms. So produce the male gametes. Second thing, it will also produce the sex hormone. So produce sperms and sex hormones. So what's the sex hormone seen in the males? Yeah, it's a testosterone. Okay. So what comes under male external genitalia? It is the penis and scrotum. Together we call it as male external genitalia. Remember, so male external genitalia, it's nothing but the penis plus scrotum. Okay. So it will include the penis and the scrotum. So let's talk about this penis. First, what does it do? It will conduct both the urine and the sperms. So conducts urine and sperms. It is containing erectile tissue, which will be helpful for transmitting the sperms into the female genitalia. So remember, it is containing the erectile tissue, which will stiffen the penis. That will help for transfer sperms into female genitalia, which is a specialized tissue. Remember this. Okay. And remember, at the tip of this penis, whatever the opening is there, that opening is called as the urethral meatus. So remember this. Urethral meatus, it's nothing but the opening at the tip of this penis. And if you see at the tip region, whatever the enlarged area is there, that enlarged area is called as the glans penis. So the enlarged portion of the penis. And this entire penis is covered by a outer sheath or outer skin. We call it as the foreskin. So covered by outer skin. We call it as the foreskin. Clear?
Yes.
So scrotum. What is the use of this scrotum? It is a sac-like structure. This sac-like structure, it is present outside the abdominal cavity. Remember, this scrotum is very, very essential. It should be present outside the abdominal cavity. So outside the abdominal cavity. And this scrotum, it was separated into two different regions. Okay, so two lateral regions. By a mid region, that's called as the raphe. For suppose if you see this scrotum, it's outside, there was a separation into two lateral sides. Okay, so one testis will be present on one side, the other one will be present on the other side. So whatever that separation portion, the middle region, we call it as the median ridge, which is raphe. Remember this. So ridge means a certain groove-like structure. Okay, so there is a cross bridge-like structure that we call it as raphe. So separated into lateral portions. So that we call it as median ridge. So this median ridge, it is called as the raphe. This is very, very important. Remember this. Clear? See, this is the external division that we call it as raphe. The same internally, whatever the scrotum is there, it was divided into two rooms, two septum. Remember this. So inside internally scrotum or scrotal septum divided into, or whatever this internally scrotal septum is there, it will divide the scrotum into two different chambers. Okay, into two different chambers, or you can say into two sacs. The chamber, we call it as scrotal sacs. So these scrotal sacs, each of them, it is containing a single testis. Remember this. So one is present on the one lateral side, the other one is present on the other lateral side. But what is the main important thing for this scrotum to be present outside the abdomen in order to maintain a lower body temperature, lower temperature? And that lower temperature is very, very important for the sperm production. Remember this. For spermatogenesis, in order to happen, you need 2 to 2.5 degrees Celsius lower temperature than the body temperature. This 2 to 2.5 degrees Celsius, it is very, very important for the spermatogenesis to happen. So remember, so this is very, very important for sperm production and viability. That means the quality of the sperms it is producing. If the sperms are not good quality, they cannot fertilize the eggs. So there is a no formation of the zygote. So in order to have a good quality of the sperm production, the temperature should be lesser than the body temperature. It is 2 to 2.5 degrees Celsius. Clear?
Yes.
And whatever these testes are there, they will be hanging in the scrotum with the help of a spermatic cord. Remember this. So what is this spermatic cord? So testes hangs in the scrotum with the help of a tube-like kind of structure. That tube-like structure is called as spermatic cord. Remember this spermatic cord, it is a tube that consists of, okay? So remember, consists of blood vessels, lymph vessels, nerves, and cremaster muscles. I'll tell you what is the use of these cremaster muscles. So for now, you understand that the spermatic cord, it's a kind of tube-like structure you can see here, the kind of tube-like structure which will be helping for the passes of these blood vessels, lymph vessels, nerves, cremaster muscle. For suppose there's a blood vessel and there is a lymph vessel and there is a nerves and there is a cremaster muscle. So all these will be packed inside a layer or inside a tube-like kind of structure that is called as spermatic cord. Okay. And remember, these testes are connected to wall of the scrotum by gubernaculum. You can see here. For suppose this is the scrotum wall. To this scrotum wall, it was connected with the help of a structure that we call it as gubernaculum. If you see, this was the internal wall of the scrotum, and this is the testis. So this testis is attached to this wall with the help of a connection that we call it as gubernaculum. Okay, is that clear?
Yes.
Yes.
So remember, testis connected to wall of scrotum by gubernaculum. Remember this. This is very important. So you can see here the spermatic cord. So this spermatic cord where the blood vessels are present, nerve vessels are present, lymphatic vessels are present, and cremaster muscles are present. Okay. So let's see the functions of this scrotum. One is it will be helpful. It is a helpful for regulating the temperature. So you can say thermoregulation. So this thermoregulation in the scrotum is brought about by three things. One is the sweat glands. We know there is a production of the sweat, so that sweat will cool down the temperature. That's one thing. Next one is the muscles. That is dartos and cremaster muscles. I'll tell you how does they will work. And the third one is palmiform plexus. So pampniform plexus. I'll tell you what is this. Okay, so remember thermoregulation, it will be done by three things. One is the sweat glands. Okay, so I told you we need to achieve 2 to 2.5 degrees Celsius. That 2 to 2.5 degrees Celsius, it was brought by three things. One is the sweat glands, one is dartos and cremaster muscle, and the other one is pampniform plexus. Remember these three names. So let's see how does individually each one will do. So we know first one, it was done by the sweat glands. Okay, so remember sweat glands. How does these sweat glands will produce? They are present in the wall of the scrotum. So remember, where does this present? They are present in the wall of the scrotum. So wall of scrotum. As they are present in the wall of the scrotum, they are present in abundant in number. Okay, so they are in large number. And all these large number of sweat glands, they will be producing the excess sweating. So there will be excessive sweating. And this excessive sweating, they will absorb the heat and they will evaporate. So this thing, this mechanism will make the scrotum cool down. Okay, so remember this excessive sweating, they will absorb the heat. So absorb the temperature and gets evaporated. So this mechanism will be providing the cooling effect. So that's how the sweat glands will be helpful for maintaining the temperature. So the next one is dartos and cremaster muscle. So what is this dartos and cremaster muscle? So you can see here the cremaster muscle here. So you can see here these are the cremaster muscles. Okay, so the dartos muscles are also just present. You can see here this is the dartos. This is the cremaster muscle and this is the dartos muscle, tunica dartos. So what does this will do? Okay, for suppose let's say if the external temperature is cool, they let's say it's winter. Okay, so what happens? There will be environment will be having the cold temperature. So in cold temperature, what happens? Whatever these cremaster muscles and dartos muscles are there, they will start contracting. When these cremaster muscles and dartos muscles, when they contract, whatever this testis is there, it will be uplifted. Okay, so it will be uplifted. When it will be uplifted, it will come closer to the abdomen. So when it comes closer to the abdomen usually will have 36.5 degrees Celsius. That's a body temperature. And these brought close to this temperature. So they will absorb the heat from the abdomen, they will absorb the heat from the body, and they will increase their testis temperature. Understood? So when the outside temperature is cold, cremaster muscles and dartos muscles, they will start contracting. Okay, so this contraction will make them move the testis closer to the body, closer to the abdomen. So when they come close to the abdomen, they will start absorbing the heat from the body. So absorb body heat. So this contraction of the muscles will cause the scrotum to become tight and wrinkled. So whatever this smooth normal skin is there, when it start contracting, it will just have a several foldings and it will become tight, it will become wrinkled in appearance. So that this contraction will prevent the heat loss. So this will act like an insulator. When it is stretched, when it is straight, when it is not wrinkled, it can heat loss. But when it is contracted, it will be having several foldings. That several foldings will prevent or it will act like an insulator layer where whatever the temperature it is observed the scrotum, it will not be lost to the external temperature. So remember. So contraction of muscles causes scrotum to become tight, wrinkled in appearance. So this will cause reduced heat loss. Is this clear? And whatever, this is happening during the cold temperature. If you put it into the warm temperature, then the reverse will happen. So whatever the events, the cremaster and dartos muscle contract, here they will relax. So they will move closer to the body. Here they will move farther from the body. They will absorb the body heat. They are far away, so they cannot absorb much heat. And now they are stretched, so there will be no contraction. That will make them tight. Okay, that will not make them tight, not wrinkle. So it will be helpful for heat loss. So that's how during the warm temperature, it will lose out the temperature easily. So warm temperature do reverse actions. Clear?
Yes, sir.
Yes, sir.
So that's one way. And the third method is by the pampniform plexus. So what is this pampniform plexus? So you can see here the pampniform plexus. Okay. So what are these plexus? Generally, this is the blood vessel. Okay. So this blood vessel, when it is branched out into multiple veins, so branched out into multiple veins, and these veins will act like a spider web. So it will form a several web-like. Okay, so that we call it as the plexus. Remember, whenever you say plexus, it's nothing but the network of blood vessels. Okay, here these plexus are called as pampniform plexus. Remember this. Okay, so this is extensive network of veins from testes to that of the surrounding testicular artery. So what happens? There is a big artery that is coming, that is bringing the blood from the abdomen to these testes. So when it's bringing the blood from the abdomen, it will be bringing the warm blood. So whatever the arterial is there, it is bringing out the warm blood. So that warm blood, it will lose its temperature into this network of veins. Okay. So remember, warm arterial blood, what it will do? Can heat up the testes. So if the testis gets heated up, what happens? Sperm production will be inhibited. So what happens then? There is a no sperms. In order to prevent that, what happened? So whatever these extensive network of veins are there, they will act like a counter current. For suppose here you are getting a warm water from this tube. So what you will do? You will surround this with a cold cloth, or you will surround this with a cold water pipes. So what happens? All that warm, it will be absorbed by this cold pipes, or it will be absorbed by the cold cloth. So now whatever that it is reaching to the final destination, this warm blood will cool down when it reaches here. The same way, here also the same thing happens. So whatever the warm blood that is coming down, because of this counter current heat exchange, it will cool down. So counter current heat exchange. So cools the warm blood. Is that clear?
Yes, sir.
Yes, sir.
So that's about the pampniform plexus. So by these three methods, the testis temperature will be reduced. Okay. So what is this testis? Testis, let's see about the anatomical features. If you see this testis, it is 4 to 5 centimeter long. That means it's very less. And 2 to 3 centimeter wide. And what is the main important role of this testis? Production of sperms and secretion of the male sex hormone. Or you can say the male androgen. And remember, if you take most of the mammals, okay, these testes are present outside the abdomen. So you call them as extra abdominal. In most of the mammals. Why? In order to 2.5 degrees Celsius lower the temperature, which is optimal for the sperm production. Okay, and remember, intra-abdomen, intra, for some, whatever the sperm, whatever the testis is there, it will happen inside the abdomen. Okay, so intra-abdominal is present in egg-laying mammal. In most of the mammals, it's outside the abdomen, so we call it as extra-abdominal. But in some mammals, which are egg-laying mammals, so usually mammals give birth directly, but some egg-laying mammals where you will be seeing the intra-abdominal. Okay, so where do you see them? One is elephants, whales, dolphins, and there is a protherians as well, prototherians. Okay, so in this, it is not present outside the abdomen, it is present in the main body cavity. Remember this. So present in main body cavity. Clear?
Yes, sir.
Okay.
Yes.
And remember, these testes in so testes of male baby, they are formed during 7th to 8th week. That means before the 2 months itself, or within the period of 2 months. Okay, so within the 7th to 8th week of pregnancy, and they will be inside the abdomen. Remember this. So once it is produced, they will stay inside the abdomen. So before birth, they will just move to the scrotal cavity. Remember this. But till then, they are present inside the abdomen. Okay. So 7th to 8th week of pregnancy and intra-abdominal. And this, whatever they are present inside the abdomen, so they will migrate to scrotum. So testes, they will migrate to scrotum during the last phase of the pregnancy. That is 7th to 8th month of pregnancy. Clear?
Yes, sir.
Yes, sir.
Yeah. And remember, this has to be moved in the 7th or 8th month of the pregnancy. Sometimes it may fail to descend into the scrotum. Okay, so that we call it as cryptorchidism. Remember, so what is cryptorchidism? So fail of testes to descend into scrotum. So we call it as cryptorchidism. Okay, and why does this failure can happen? Due to the deficiency of the testosterone. So deficiency of testosterone. Clear?
Yes,
Yes,
sir.
sir.
And now where they are present? They are present inside the abdomen. Inside the abdomen, the temperature is very high. So will they able to produce the gametes? Will they able to produce the sperms?
No, sir.
Yeah. So that will cause the sterility. So causes sterility. Okay. Because no optimal temperature. So whatever the 2 to 2.5 degrees Celsius lesser you want, that optimum temperature cannot be maintained. So no optimum temperature. So they will cause the sterility in the males. And remember, whatever the cryptorchidism is there, 15% they will just be present in the abdomen, 25% they will be inguinal canal. So that's the canal which will connect the abdomen to the scrotum and eye scrotal. That means just above the scrotal cavity they are present in 60% of the cases. Okay, and what we can do, the doctor can do the surgery and he can move whatever the test is present here into the scrotum. So that surgery name is called as orchidopexy. Okay, which is the surgery performed to correct this cryptorchidism. Okay, remember this. So surgery to correct cryptorchidism. Here, understood?
Yes, sir.
Yes, sir.
Yeah. So yeah, so male reproductive system. If you say the male reproductive system, it was classified into three types. One is the male external genitalia. There is a male internal genitalia, and there is a accessory ducts and glands. So remember, so one is the male external genitalia. The other one, it is the male internal genitalia. And the third one, it is accessory ducts and glands. Okay, so what comes under male external genitalia? Just now we saw. One is,
Scrotum and penis.
yeah, one is the scrotum and penis. So what will come under male internal genitalia? That's nothing but
Yes.
testes. And what will come under accessory ducts? It's nothing but the sperm ducts. We'll see. The epididymis, vasa efferentia, vasa differentia, all those, reti testis. So sperm ducts and glands. So will comes under accessory ducts and glands. Clear? So let's see about the anatomy of these testes. So far we saw the external features. Let's see about this anatomy. So first of all, if you see these testes were divided into multiple compartments. You can see this is one lobe, this is another lobe, another lobe. Okay. So remember each testis, it was divided into a multiple chambers. Those multiple chambers, we call it as the testicular lobules. Okay. And these testicular lobules, they will contain almost 250. So remember, each testis has about 250 compartments. So those 250 compartments, we call it as the testicular lobes. Clear? And now each of these testicular lobules, so whatever these testicular lobules are there, each lobule, okay, so there are total 250 lobules are there. In that each lobule will contain three highly coiled seminiferous tubules. Okay, so each lobule contains 1, 2, 3 highly coiled seminiferous tubules. Okay, so remember this. So in this 1, 2, 3 highly coiled seminiferous tubules where the sperm production happens. Okay, remember. So in testis, where does exactly the sperm production happens? It happens inside the seminiferous tubules. Remember this. So here, sperm production happens. Clear?
Yes, sir.
Yes,
And
sir.
if you see here, this testis, it was outside, covered by a layer. That layer, we call it as the tunica. Okay, so this tunica, it is of three layers: outer layer, middle layer, inner layer. So the outer layer, we call it as the tunica vaginalis. The middle layer, it's a tunica albuginea, and inner layer, tunica vasculosa. Remember this. Okay, so tunica, which is nothing but the protective covering of testis. So this protective covering of the testis, it was made up of three layers: the outermost layer, the middle layer, and the inner layer. So what do we call this outer layer, outermost layer?
Tunica vaginalis.
Yeah, it is a tunica vaginalis. The middle layer, it is tunica albuginea. And this one, it is nothing but vasculosa. So you can remember like VAV. Wow. Okay. So remember this. Tunica vaginalis, tunica albuginea, tunica vasculosa. So next one. So you can see here, these are the seminiferous tubule compartments, and this is the seminiferous tubule. So this seminiferous tubule, it was about 70 centimeters long. Sorry, it was about 70 millimeters long. Okay. And remember, each seminiferous tubule, it was lined by two types of cells. One is these cells, the other one is the serotolite cells. Here. For suppose if you see here, this is the round. Okay. So this round, it was having a single cell like this, a single cell like this, like this. Let me take only four. So like this. Okay. So between these single cells, there will be multiple cells are arranged. So you can take like this. See, these are not boxes, it's a single cell. Okay. So for suppose, let me take a nucleus inside. This is nucleus, this nucleus, this nucleus. And outside, there is a cells or multiple cells were present. So this single cell, okay, so whatever this single cell is there, it is called as the serotolite cell. So you can see here, this is serotolite cell. This is serotolite cell. So this is serotolite cell. Okay, and between these serotolite cells, whatever the cells are there, the orange color, so they are called as the spermatogonia. Okay, so these are spermatogonia, and this one, it is a serotolite cells. So what's the use of this spermatogonia? So these are spermatogonia, they are nothing but a male germ cells. Okay, so they are immature male germ cells. So they will mature and they will become the sperm, spermatozoa. Okay, so they are called as spermatogonia as they are immature. That will turn out to be spermatozoa. That's nothing but a sperm. And serotolite cells, which are present as a single in between, so they will helpful for nourishing the germ cell. So nourish the germ cell. So you can see here, this is one seminiferous tubule. This entire thing, it is seminiferous tubule. Outside this seminiferous tubule, whatever this space is there, that space we call it as the interstitial space. Remember this. So whatever the outside space is there, that we call it as the interstitial space. So what is this use of this interstitial space? So this interstitial space, it is outside the seminiferous tubule, and they contain a kind of cells called as the Leydig cells. Remember this. So these contain, or interstitial space contains Leydig cells. So what's the use of these Leydig cells? Yeah, they will synthesize and secrete the male sex hormone, which is nothing but the androgens. And the other one is the internal one, which is the serotolite cells. Okay, so we know the serotolite cells will be helpful for, one is it will give the structural support for the inside seminiferous tubule. Okay, and the second one, they will produce the nutrition for the sperms, and they will also helpful for protection to the development sperms. So three functions will be done. So remember, So this one, we call it as serotolite cells. So these serotolite cells will be helpful for structural support, nutrition, and protection to development sperms. So protection to the developing cells. And remember, they are just present extending to the basement membrane, okay, to the lumen. For suppose this is the basement membrane. From this basement membrane, it was extended towards the center. So the center you can call it as the lumen of the tubule. Okay, so remember, it was extend from basement membrane to lumen of tubule. Clear? So spermatogenesis, this is the nothing but the process of production of the sperms in the seminiferous tubules. Okay, so we call it as the spermatogenesis. This will happen once the person reaches to the puberty. Okay, so before puberty, the spermatogenesis doesn't take place. Okay, so tell me what is spermatogenesis? It's a process of formation of sperms. Okay, remember, it's a process of formation of sperms. When it will start? It will start after puberty. Okay, so begins with during puberty. Once the person reaches to the maturity, it will start begin. Okay, and when it will stop, it will never stop. It will continue till the person die. Okay, so there is in females, it will happen till to the some time. Okay, after that it will stop. That we call it as menopause. But in males, it will not happen like that. It will continue till the death. Okay, but the sperm quality and sperm number may reduce with the age, but continues till death. As the person ages, the sperm quality and the sperm number will reduce. Okay, so remember this. So with advance in aging, so number and the quality of sperm reduces. Okay, and this spermatogenesis, it will be classified into two types. Okay, I mean it will be classified into two stages. One is spermatocytogenesis and spermiogenesis. Remember this. So one is spermatocytogenesis. The other one is spermiogenesis. So what is this spermatocytogenesis? That's nothing but formation of the spermatids. So formation of spermatids. So as it is forming the spermatids, it is called as spermatocytogenesis. Okay. So from where these spermatids will form? From spermatogonia, or you can say the immature germ cell. So from spermatogonia. And what is this spermiogenesis? So whatever this spermatid it is formed, it has to convert, it has to mature into a mature sperm. It has to develop into a mature sperm. So that we call it as spermiogenesis. So that's a differentiation. That means development of spermatids into sperms. Clear? So remember, spermatogenesis, it is classified into two types. One is spermatocytogenesis, that is formation of the spermatid, and the other one is spermiogenesis. That means whatever the spermatid is formed, it has to develop into a functional mature sperm. So that we call it as spermiogenesis. So first spermatocytogenesis will happen, and later the spermiogenesis will take place. And remember, how much time does it takes for the spermatogenesis to happen? So spermatogenesis, it takes at least 65 to 75 days. Remember this. You can see here. First, initially there is a spermatogonia. This spermatogonia, it has to form a lot number of spermatogonias. Okay, it has to increase a lot of number. In order to increase this lot of number, it will undergo mitosis. So the single spermatogonia or the single sperm germ cell, it will convert into sperm mother cell, it will convert into many. So once the many are formed, now it will undergo meiosis 1, first meiotic division, and it will form secondary spermatocytes. So suppose if you see here, it is 2n, it is 2n, it is 2n. But after first meiotic division, whatever the 2n is there, it will now become 2n. Remember this. Okay. So tell me now, how many cells, the primary spermatocytes, it will undergo the first meiotic division and result in formation of haploid cell. So secondary spermatocytes, it's haploid in nature. The primary spermatocyte, it's a diploid in nature. Now this further undergoes second division and it will form the spermatids. So these spermatids will undergo maturation and will form the sperm. Clear? Or it will
Yes,
undergo
sir.
differentiation. So remember this, whatever this spermatogonia is there, or you can also call this spermatogonia as a germ cell. So it is also said as the sperm germ cell or sperm mother cell. Okay, so this will undergo multiple mitotic divisions and it will form primary spermatocyte. Remember this. Here, multiple mitotic division will happen. So mitotic division is mitosis will happen. Result in formation of the primary spermatocyte. So this primary spermatocyte, it will undergo first meiotic division or it will undergo meiosis 1. So once it undergoes meiosis 1, whatever the diploid cell is there, here it's still diploid, here it is still diploid. Now this diploid cell will become haploid. So it will form the secondary spermatocyte, which is haploid in nature. So it will continue 23 chromosomes here, 46 chromosomes here, 46 chromosomes. Once meiosis will happen, it will get 23. Now this will undergo further meiosis 2 and result in formation of again the haploid cells, which is nothing but the spermatid. Okay, so how many are produced? 4 spermatids are produced. Remember this. So it will produce 4 equal haploid spermatids. So 4 equal haploid spermatids. Clear?
Yes,
So,
sir.
Yes.
so finally, whatever the spermatid is formed, that spermatid, it will become the functional mature sperm. So that we call it as the spermiogenesis. So within the spermatogenesis, it will happen. Spermiogenesis. Okay. So what happens here? Differentiation will happen. What is that means? So for suppose appearance of the acrosomal, so you can see here, initially this is present like this, but there is a acrosomal vesicle and flagellum will start developing and the acrosome will just grow further and flagellum will grow further and then shedding of the excess cytoplasm, whatever the excess cytoplasm is there, it will release out of that and then it will become completely functional mature sperm. Okay. So this entire process, we call it as spermiogenesis. So once it will form the mature sperms, okay, after spermiogenesis, whatever the sperms are there, so these sperms, or you can say these sperm heads, they will become embedded in Sertoli cells. So whatever the Sertoli cells is there, okay, in the round, the Sertoli cells are there. So they will just put their head into this. They will embed their head here. Okay, so embedded in Sertoli cells. So this will form the specialized sperm. So forms the specialized sperm. Now this specialized form, it will become fully functional and they will be released out of the seminiferous tubule. Okay, so sperms finally released into, or you can say released from seminiferous tubules. So these sperms which are releasing from the seminiferous tubules, so they are called as spermiation. So remember this. So finally spermiation will happen and the sperms will liberate out. So that's about the spermatogenesis. Any doubts?
No, sir.
Okay, then I'll just send this PDF. Once again, read it.
Sir,